Good morning. Today we will be describing to you briefly the epigenetic effects of alcohol on an infant in utero.
So firstly, most of you have probably heard of the epigenome, which literally means above the genome. Through methyls and histones, the epigenome effectively tells the genome what to do. Recent studies have shown that the epigenome may potentially be connected to things like determining personality and reactivating tumor-suppressing genes. Most importantly, the epigenome of an individual affects physical and mental traits in following generations.
It is commonly acknowledged that drinking during pregnancy increases the risk of birth defects, and the questions we have been investigating are why and how.
Epigenetics and evo-TiVo are relatively new fields of study. We will be demonstrating epigenetics by focusing on FAS, or also known as fetal alcohol syndrome. More specifically, we will be talking about FAS-induced neurological developments.
When we talk about drinking, we mean any kind of drinking. When it comes to drinking during pregnancy, there is no moderate dose, as we do not have enough knowledge about epigenomics. An example is diminished neural response in children birthed from mothers who drank during their pregnancy. This can be in very slight terms, such as mildly diminished learning ability, strange emotional displays, etc. Yet there's a definite correlation.
FAS is, in a sense, a prime example of epigenomics. In its embryonic stage, a human is most vulnerable to changes in its environment, that is, the womb. And a mother's drinking, which is the cause of FAS, completely changes the environment because approximately 30% of foreign substances, such as alcohol, are able to cross through the placenta into the infant's bloodstream where they can wreak havoc. Here we have a diagram showing some of the many effects of FAS. Mental consequences. Here is my colleague Kevin to explain them to you.
I will be focusing on a specific disorder called microcephaly. Microcephaly is a common effect of fetal alcohol syndrome. It is characterized by a small head size and a small brain size. However, the face of the fetus tends to develop normally. During early development, the head does not form properly, which usually leads to mental retardation. Here is a diagram of two babies. One has microcephaly, the other is normal. As you can see in this diagram, the baby with microcephaly has a much smaller head size than the baby without microcephaly. However, both faces seem to be developing at a normal rate. Since alcohol is not a mutagen, these effects should not be due to changes in the actual genome. These effects are actually due to epigenetics, which are changes in the gene expression, not the actual sequence of nucleotides. Tests on frogs have shown how these alterations work. Alcohol was injected into developing fetuses, which showed reduced expression in genes that were used to develop the nervous system. One gene in particular, XPAX6, was suppressed up to 90%.
Along with these, there are a variety of other nervous disorders that are attributed to FAS. Among these are learning disorders, speech and language problems, delayed reaction times, hyperactivity, motor skill problems, and these are primarily attributed to the fact that drinking alcohol lowers the pH of the blood of both the mother and her offspring. In one study, this was shown to decrease the levels of one certain type of brain cell by up to 24%, and this permanently damages the central nervous system of a child and permanently alters their epigenome.
Research found that maternal drinking was causing decreases in maternal thyroid hormones, and therefore a decrease in thyroid hormone levels within the infant. In the fetal brain, there is an enzyme that controls thyroid levels. This enzyme, abbreviated DIO3, is epigenetically imprinted for production on the gene. This usually comes from the father's genes, while the mother's genes for DIO3 are silenced. However, alcohol disrupts which genes are silenced or not, causing abnormal doses of thyroid hormones within the fetal brain. These zones of the brain are all connected to learning, behavior, emotion, etc. Although correlations between the epigenome and diseases or deficiencies are very new and unexplored, it opens many avenues and gives scientists the opportunity to explore new cures working at the root of the problem.